Everyone can hear. Hopefully everyone can see my screen. Let me know if you can't. Great. All right. Well, welcome everyone. I'm Monica Poelchau. I am one of the AgBioData steering committee members and one of the co-PIs of the Research Coordination Network grant. And I'd like to welcome everyone to our third community workshop, which is virtual this year. So thank you very, very much for coming. I wanted to start by going back a bit and reminding everyone what our Research Coordination Network grant was about. I'm sure everyone's seen this before and has heard this, but it would help. I don't think it would hurt to refresh everyone's memory about this. So our goal with this RCN was to really expand our existing AgBioData network that started back in 2015 and was basically unfunded or almost unfunded up until then. Just to expand it, create an inclusive network amongst genetics, genomics, and breeding databases, researchers, scholarly publishers, and funders to really dig into FAIR data and help implement it across the board, to help with education and encourage the use of the standards that we will develop, and to think about the future of AgBioData and its sustainability. So to that end, we have 4 aims, and we'll hear a lot about the progress on those 4 aims in the next couple of days, I'm sure. The first aim was really sort of a grassroots approach to identifying what problems or gaps might exist in the work that we do in genetics, genomics, and breeding databases, and to come up with, what to find those gaps and come up with possible solutions to help us move our work forward. And that was in the purview of working groups. Our second aim was to expand our network to include key stakeholders. So those are those researchers, scholarly publishers, and funders that we mentioned up here above. Our third aim is to really work with our stakeholder community and others to help educate on FAIR data, excuse me, but also to help communicate what we databases do to help stakeholders make their data more fair. And our fourth aim is to think about sustainability of our network and of the databases that are parts of our network to help all of our databases to become financially sustainable in the long run. I wanted to step back a little bit. So we're in the 3rd year of this RCN, and I'll talk a little bit about that in the next slide. But what we've already accomplished. So we have 197 new members since the start of the grant, which was in September 2021. So that's a 230% increase. We have 7 active and 7 completed working groups, 4 sets of recommendations for databases, including 2 published papers, 6 workshops, so 1 training workshop, 3 community workshops where we all come together, including this one that we're at right now today, and 2 workshops at the Plant and Animal Genomics meeting. We have education workshop materials, which we'll hear more about during this workshop, 26 monthly webinars, 9 presentations and 3 posters on AgBioData's work. And that's just not to mention even all of the work that we're going to hear about today that is still in progress, starting, or just wrapping up. So there's lots more good stuff to be added to this list. So I wanted to step back a little bit and say thank you to everyone here on this call, who's not on this call but contributed to this work previously, because this is all, AgBioData is all about you and all about this community's contributions to this network. So we would not be anywhere where we are without the community's work. So give yourselves a hand, a pat on the back about all of the time that you volunteered and worked to make AgBioData where we are and a better thing. I wanted to then go back and explain a bit about where we are in the context of timing of the RCN. So we, as I mentioned before, our funding period started in September of 2021. And in the first year, we had our first tranche of working groups launch, our first community virtual workshop. We started out with sustainability surveys. Second year, each year we iterate around those working groups. And so we had some close up, some begin. We had reports from the year 1 working groups. We had our Chicago workshop, our in-person workshop in Chicago. Year 3, we're about here right now. Again, we're going to hear about new working groups that launched this year, about ones that have closed out. We have our third community workshop right now, and we'll hear about the education curriculum. And this is just sort of a high level, tease about what we've actually accomplished, just to remind ourselves. So technically speaking, the 3-year funding period would end sort of at the end of 2024, but we've talked about the possibility of continuing this work with a creativity extension. And thanks to NSF, we actually can really talk about the creativity extension because it will happen this fiscal year. We received the go-ahead. So thank you to the National Science Foundation first for funding this network initially and the RCN, but also for really acknowledging the work that everyone in this network has done with this additional funding pulse. So we really, really appreciate it, and it's an honor to be able to receive this. We are aiming for 2 additional years. I'm not going to delve into exactly what we're doing because it's still, we're almost at the end of forming and submitting that, but we still want to just cross our T's and dot our I's. But essentially, we want to continue with the great work that's been done among those 4 aims so far, and with some slightly different aims for encouraging people to continue on. So stay tuned for those details, but that's our current aim. If you have any questions about that, you can reach out to the writing group, which right now is Lenore Reiser, Carson Andorf, Dory Main, John McNamara, and myself. And thanks to everyone already for the input and feedback that you've given as we've solicited feedback from this group on the creativity extension. We really appreciate it. Okay, so now I need to talk about what we're actually doing in the next 3 days with the community workshop. So as we've done in previous workshops, it's pretty similar, but the goal is to hear from the working groups, share progress and outcomes, and to help those working groups further what they're doing with the feedback and insights from the larger group. And so this will happen on all 3 days, Monday, Tuesday, Thursday, via presentations and breakout groups. On Tuesday, we're going to do something a little bit different for us. So we want to recognize the work that's been happening within the community, and we have a sort of awards and recognition ceremony then. And on Thursday, we typically in these workshops also talk about what our future. And so we have an additional discussion scheduled for Thursday at the end of that session on the future topics for AgBioData. So please attend that as well. So really briefly about the agenda. I think we all received a link about that, but I'm going to share that here on this webpage. So today is our first day. After I finish talking, we will hear from 3 different working groups, which Peter will introduce in just a minute. We'll have a short break, and I encourage everyone to stay on because after that, the important work in the breakout rooms will happen where we'll all convene, figure out what group we're going to go into, and focus on questions that the working groups have defined for us. And then there'll be short reports in the breakout rooms and final remarks. And that sort of narrative will continue on the second and third day. I want to remind everyone of our code of conduct. You can find the link here. I think Anna-Rita shared it again last Friday. Essentially, be respectful and nice, but feel free to read through it if you can. There will be presentations first, as I mentioned, in breakout rooms, and there'll be logistics, a little primer on logistics in that right before the breakout rooms on how that will work. If your breakout room lead asks for a scribe or a moderator and they don't have one yet, please feel free to volunteer for that. There'll be technical help in this main Zoom room via Anna-Rita if you need it. And then we also have a Slack channel if you need to get support there. All right, and before I turn it over to Peter, I just wanted to mention here's our current steering committee. Some faces may have changed since the last time you saw us here. So if you have any questions or comments, please feel free to reach out to any one of us. And I want to give a big shout out to Anna-Rita Marano, our coordinator, who's the driving force between all these things that happen here. So big shout out to her, and also Sunita and Meg, who have been the steering committee leads on getting this workshop going. So thanks to all of you. Okay, and with that, I will stop sharing and I will hand the baton over to Peter Selby.
Awesome, thanks, Monica. Yeah, we'll get started here. I'm Pete Selby, new member to the steering committee. So yeah, thank you for having me. I'll be your moderator and MC for today. I'll be keeping track of time as well, so I'll be the one cutting you off if you present for too long this morning. So without further ado, we'll get started with our first presentation. This is coming from Ethy. This is going to be our first working group that's closing this year. The Genome Assembly and Annotation Nomenclature Working Group is ending this year. And with that, I'll turn it over to Ethy.
Okay. Oops, wrong one. Hey. So I'm Ethy from Ames, Iowa, USDA. I'm going to give this presentation on behalf of the Genome Assembly and Annotation Nomenclature Working Group. So this is problem. Naming of things matters, but naming of genome assemblies, annotations, gene models is pretty out of control, which makes it really difficult, among other things, to do an analysis across multiple genome data sets. And you can spend anything from hours to weeks trying to get the names all in line so that you can run a pipeline or an analysis. And then, of course, then you need to cross-reference to get back to the original names. And then just kind of knowing what you're dealing with, even if you're doing individual genome assemblies or gene models. And then you get cases where different groups give different names to the same thing. And often I've observed, not always, but often researchers get so buried in their work, they don't realize the names matter, or because they understand their names, they figure that they are the best possible names, or underestimate the difficulty of developing a nomenclature that works in a generic sense. So what's the solution? Is there a solution? Change is really difficult, and standards are difficult to enforce. And there's no nomenclature that's going to be perfect. It's just the problem is too complex. And so you're always going to get people who dislike what you've got and they'll try to improve it. And then you get different research cultures, for want of a better word. They have different approaches. It might be, literally cultural differences. More likely it's a community difference. Gene by gene researchers are going to want one kind of name, and people who do large analyses across multiple data sets are going to want a different approach. People who want doing AI might have a different approach. And then different organisms can have special needs, maybe dealing with polyploidy or doing haplotype assemblies or organelle assemblies, or who knows what all. So I think we are the oldest existing working group. We were formed right at the beginning of 2021. And the fact that we're just now wrapping up, I think, is an indication of how challenging this problem is. And we have finalized the white paper, which I hope to get submitted to bioRxiv within the next few weeks. And here are current active members. And as you can see, we've got a pretty good spread across different types of organisms represented here. Sarah and Kapil are the co-chairs, but they have done many presentations, so I volunteered to do this one this time. So the long and short of what we have come up with, and what we're recommending. First and foremost, it's very important that people understand that nomenclature matters. You can't just slap a name on a thing and call it good. Use your community's nomenclature if there is one. And if you don't like it, work with the community to fix it. Don't go off and come up with the perfect nomenclature on your own. These nomenclatures need to be both human and machine readable. And gene models should identify the source assembly. I've frequently had a gene model or a handful of gene models and had no idea where they came from. And then if you are in a position to create a new nomenclature for your community, consider using our recommendations. So we started off by looking at what already exists, and we found quite a few papers on nomenclature rules, but they apply typically to genes in the classical sense, loci, not a gene model, repeats, proteins, strains, so on. little to nothing addressing the names of assemblies, annotations, and gene models. But we did find a few formalized nomenclature standards. One that I'm very familiar with for maize. This has been around for 10 or 11 years, and we've had really good adherence to it. Not perfect, but very good. So assembly name, this is intended to be very human readable and machine readable. So genus, species, one letter for each. Short cultivar name or accession, the reference with the quality, which could be, say, reference or draft, the group that did it, and the version number. And it's a major dot minor version number. But this is sort of a long cumbersome thing. So we have a shorthand, genus, species, again, 2 letters, assembly ID, which is a number, and these are assigned in the order in which the assemblies are created. A version number, which is a letter, not a number, and an annotation version, which is again a letter. And so the gene model has the short identifier followed by a 6-digit gene model number. There's examples of that. Another one that takes a very different approach. This one's been around, I think, six years or so. The Legume Information System takes in genome assembly and annotations from many different sources. It's they're not in a position to enforce any sort of nomenclature on the community. So instead, they do this. So that to identify an assembly, they use what they call a gens indication, which is the 3 letters of the genus, 2 letters of the species, dot cultivar, dot the letters GNM, meaning genome, and the assembly version, and then a 4 alphanumeric identifier randomly produced. And this identifier serves as the key to the metadata. So if you've got this identifier, you can get to the metadata. And the original genome name and its source and lots of other stuff is listed in the metadata. And so for an annotation, use all the assembly stuff plus .ann and the annotation version, followed again by that identifier. And for the gene model, they take the annotation as a prefix dot, and then they use the original gene model identifier. So in some sense, they're renaming these gene models, but you can still get to the gene models using the original gene model name that was given by the group that did it. Okay, a brand new one. This one's for Apple. Works this way. There's the long, friendly, human-friendly name. We have the species, sample identifier, consortium. In this case, that's called ABC, and the version, just major version dot minor version. But then there's a short assembly name. This uses the tree of life ID, which I'll talk about in a little bit. Short species, sample identifier, consortium version, subversion, and optional. You've got anything extra that you need to tack on to the end. For example, this could be the original assembly name if it had come in from outside. And then the gene model takes the short assembly as a prefix, the letter A, the annotation version, a single letter indicating the organelle, the chromosome, and entity, gene, transcript, what have you, and then the gene number, and if you need to, something optional tacked on to the end. And here are some examples of those. So here's what we came up with for recommendation. So the genome assembly name, that should have these parts: species, the cultivar, accession, individual, strain, what have you, the assembly version. And we had hoped to include the group or the consortium that did the work, but this ended up not making its way into our final recommendation. But could be hacked on to the end of the name. So the tree of life IDs, these are guaranteed to be unique for every species. And this starts with two lowercase letters, one for the taxonomic rank, one for the clade, and then 3 letters of the genus and 4 letters of the species. These are maintained at the Tree of Life website, and if by some chance your species does not have a name, you can request one for it. So here are some examples. So the maize version 5 assembly could look like this, and there's a Brassica napus, and Malus domesticus honeycrisp. And this last one we'll continue on as an example, and I like to point out this is a haplotype assembly. So gene model naming, assembly name, annotation version, chromosome identifier, entity type, and then a 6-digit identifier. The annotation version in this case is an integer, a single integer, no subversions. Chromosome identifier is fairly open, so it could include the subgenome and or a haplotype. And examples of entities, one letter, could be a gene, pangene, transcript. I'll come back to pangenes in a bit, so that's a bit problematic. And then the 6-digit identifier could be random, it could be ordered, it could have gaps. If you think you are going to fill in the gaps with listed gene models, for example. So here are some examples of what this would look like. So there is some a maize gene model, Brassica, apple. This, and here, as I pointed out, the Honeycrisp is a haplotype, so the hap1 becomes part of the chromosome name. And then for isoforms, instead of a G, you would have a T and a dot number for the number of the isoform. Okay, pangenome nomenclature. So this is something that got talked about a lot with different groups, and it's quite challenging. Partly, it depends on how you define a pangene, and what is a pangene? What do you hang an identifier off from? From which do you hang an identifier? But people really want names for pangenes, so we came up with some ideas. None of these are particularly recommendation. So you could have, say, clade version pan and a 6-digit number, or clade version. Some pangene analyses calculates sort of a consensus position. Maybe that could go in there, or a consensus chromosome, although obviously this isn't going to work unless you have good chromosome to chromosome correlation within your clade. Or you could have a clade group version pan number. But I guess the key here again is be consistent and follow what your community is doing. So a summary of our outcomes. Final draft will be submitted by our archive in the next week or two, and that will be followed by a submission of our white paper for formal publication. And we will also submit a letter emphasizing the importance of using standard nomenclature, and hope that these two can go together as a pair. So this group's been around for a long time, so there are a lot of people who have served on the community, and the people with stars are still active. That's it. If I went
Awesome.
over, there's time for questions.
We've got time for one, maybe 2 questions. Thank you very much. Got a question from Rex. Do you want to ask it in person?
Sure. The first question is, who decides if an assembly is reference quality? And then the second one revolves around the gene names. Which form of the gene name should be used in a publication? The original name or the fully qualified name? And would a table and supplemental data with the conversion do?
Okay, the first question is pretty easy. If pseudomolecules have been generated, at least in maize, that's our definition of a reference. And if it's in pieces, then it's a draft. The second question, which I suddenly have forgotten.
form of the gene name should be used?
Right, okay, form of the gene name. Yeah, we talked about this a bit and thought, well, could you, because I know that with good reason, people hate putting those really long gene names in papers. And we had talked a bit about suggesting that people could strip off some or all of the prefix. And that's a bit challenging. You probably could. One concern was that if you're trying to parse the paper with a an AI tool or a text mining tool, it may not know how to interpret the stripped down versions of the name. But well, for example, in the case of the LIS names, I think that it would be safe to strip off all of that prefix, since you can still get to the gene name. So it's a bit of an open question.
Well, in theory, the paper would have the information needed, what assembly was used and, what was the genus and species. So it might be there in the paper, even if it isn't on every single gene name.
Yeah. Yeah, and as I said, we kind of went back and forth between recommending that and not recommending it. So I think what we're doing is leaving it to people to decide. But keep in mind, can it be parsed by a text mining thing, and can the gene names be reattached to where they belong?
Okay, thank you.
All right.
Awesome. Thank you, Ethy. That's all
Great.
our time. So yeah, now I'll invite Megan O'Donnell to represent the education working group. And yeah, this is a currently active working group. And so yeah, take it away, Megan.
Okay, just a moment. Hi, everyone. I'm here representing the Education Working Group, and we have some exciting updates to share. First, a bit of an overview on who we are. Our membership has fluctuated quite a bit since we started. The members in yellow are currently active. The ones in white, our former members, are currently inactive members who have had to either drop off due to conflicts or due to our meeting times. I do want to point out we are currently all or mostly in North America. The Anna Rita did just move, so we do have another blip on the map that's not up to date. And an overview of our mission is that we are aiming to create an educational curriculum that teaches fair data principles within the context of GGB agricultural research, which sounded very simple until we started breaking it down. But some of the main goals are to kind of explain the purpose, structure, and uses of these databases, how the FAIR and trust principles both are really important for how these databases work and contribute to the scientific community and to reproducible research, how to access, find, and use the data in the databases, how to just find, use, and make fair data, and how the databases, along with fair data in general, advance scientific knowledge and global progress. So some of the reasons the education side is important is that this does build skill development for future researchers and current researchers. This is complex science, which requires complex systems and increasingly large and complex data, often multiple types and formats. So you have to learn different literacies to just be able to do science. It's not enough to just be a scientist now. You also need to be a little bit of a data scientist. It helps with recruitment. Researchers, curators, and educators learn to use these databases. That means more users, more scientists, and new community too. And it does also communicate the values of this community through both ethics, scientific, and technological community values. So ethical fair data fits right into that. Scientific, the standards Ethy just talked about are really important. Technological, that goes into the trust principles, which we'll talk about a bit later. It also creates policy awareness. Sustainability is a big issue as well, where we really try and emphasize that the quality of the data that these databases steward is really worth the cost, both time and money that goes into them. And just good practices, because you don't want garbage in and garbage out. You want good data and better science. So this year we really tried to refocus and refine our purpose. And we do know that the fair data principles as they relate to databases is not covered in the current education literature out there. There's a lot of fair data education, but a lot of it is focused on just general principles and not as applied to databases. So that became our big focus. And we do know that the fair data is a uniting principle for all of our databases, but the methods to achieve fair data through a database are usually hidden to users because they're technological. They're through the standards we employ, the naming conventions, the system design, and your users need to be literate in all of these things to understand how the databases are doing this. And so kind of servicing these hidden methods is a big reason for this curriculum. So our introductory lessons are focused on the FAIR principles: findable, accessible, interoperable, and reusable. And I want to just emphasize that as databases, the strengths are really in your systems and your standards. You become an authority as a database to be able to say, this is what we require, this is what we need. And if you don't follow that, it doesn't work with our system, and it doesn't work with other data. And so that is a big reason these databases are so valued. And we really wanted to make sure people understood why that is. And we also kind of refined who our audience was. We developed some very basic materials for high school students, more introductory materials for the undergraduate, and then added some deeper readings and activities for graduate students. At first, we tried to include high school students in all the lessons, and that became a bit too ambitious for some of the lessons that required more deeper knowledge of some of the science skills than we thought might be appropriate. We really focused on the conceptual and applied skills, also just how useful and important this information was going to be. And our outcomes, we want them to be open access and multimodal and easy to use and remixed by others, so that we're not the only people providing this and maintaining it. We want others to also add to it and build off of it. So in brief, our core curriculum establishes the basics. What is a biological digital repository, or a database, or knowledge base? There's lots of different names, and that's one of our big part of the lesson, is that the naming is a little complicated. How FAIR and databases work together, what biodatabases are and how to use them, creating and sharing trustworthy data, submitting data to a database, using library resources and database for agriculture. Or this is maybe the short version, which is, oh, that's what databases do. Hey, standards are important. I can use this. This is valuable. I can get help, and this work has impact, which are kind of like the big high-level summaries of why we chose these lessons as our core curriculum. So the core curriculum is finalized, and the slides and lesson plans are complete. The recorded lectures are partially complete. We have a white paper that we're finalizing the text for. A virtual workshop at Plant Biology 2024 is upcoming next month. And we have, or sorry, well, it's almost May, so almost next month. And publication of teaching materials is also upcoming after we finish that white paper and the lectures. All of these materials will be online, and the white paper will link into the teaching materials and vice versa, with a more broad overview and historical framing. One thing to note is we do know that this is just a core set of lessons. It is not everything. And we have identified some other areas for potential development. Last working group meeting, an introduction to ontologies was emphasized as a really important lesson, and we would like to add it. Database technology, both basic and advanced, we also view as important, as well as how to do more specific activities that relate to specific databases, organisms, and methods. However, we need new volunteers to make them happen because we don't all have the literacies to do these lessons ourselves. And so if you are interested in volunteering and developing these lesson plans, please contact us. We would like to hear from you and would welcome you to our group. And we want to emphasize that anyone in AgBioData, once these are published, can link to, remix, and reuse the materials to educate the users of the databases. So some of the recommendations we have for AgBioData is to create a centralized open access sustainable repository for working group outcomes. We're looking at Zenodo for ours, but it'd be great if the whole of AgBioData had a centralized one. We also need to recruit new working group members from different communities because we're overrepresented by plant people and librarians. I'm a librarian, by the way. So if I'm not going to some of the technical details on the databases, that's why. We also could use representation beyond North America and R1 universities to make sure that our educational materials are covering all the literacies at the appropriate points. And we could use more educators who can help us adapt and improve materials for different audiences. I said we get back to the trust principles, and this is where I just want to emphasize that each database is responsible for providing instructions for users. These lessons are not trying to replace that, but they do want to build off of it and help. And I would really encourage everyone who has a member database to make sure you are communicating not just the fair data principles, but the trust principles, which we cover in lesson 4, because this is what establishes your service as transparent, responsible, user-focused, sustainable, and using technology in a responsible way and appropriately. And this really does help you meet funder requirements as well as inform and educate your users to make them better scientific citizens. Here's another slide with more information on the trust principles if these are new for you. I'm not going to go over them, but you'll have access to the slides. And thank you. That was an overview of our work to date.
Excellent. Thank you, Megan. Are there any questions, comments for Megan and the Education Working Group? Yeah, Jackie.
Hi, great presentation, Megan. You mentioned that the recorded lectures will be available soon, and that like the documentation for those lectures are ready also, or not?
So we have the lesson plans, which have kind of the core concepts that are being taught, notes on how to teach it, especially if there's an activity. That part is finalized. The slides also have teaching notes on them, and then the lectures will be explained in the curriculum notes, I think. Sorry, did I answer the question? I was trying to go over all the parts we had.
And then when are these available for us to start linking to our individual databases as community resources?
Not yet.
Not yet. Okay.
That is the forthcoming part. We're working on finalizing the recorded lectures, and then we're going to publish the white paper and the curriculum all at once, I believe is the plan. If Meg or Anna Rita is on here, they can back me up on this, or maybe Rex can too.
Thank you.
Yeah. Rex, you got a question or
Yeah,
comment?
have you actually approached any faculty about including any of these materials in their curriculum?
So we do have a Meg is on this call. She is a teaching faculty. Do you want to answer the question?
Yeah, I've incorporated the first 4 lessons into a 3 credit hour graduate course on bioinformatics, which I think it's important to think about FAIR and trust in databases in terms of bioinformatics. And it's a great opportunity for the students to, one of the projects I have them do is write their own data management plan for their master's or dissertation, which is a great exercise. So it's been very successful for me, but I've been involved in the committee, so I'm a little biased. I have had a few people request that I send them links, but haven't gotten any feedback on how it went. So if anybody would like to teach with it and give us feedback, we would love that.
Thanks.
Awesome,
If
thanks.
you know educators who might be interested, please let us know. That's part of the part we need help with actually is making those connections. Word of mouth is always the strongest way to pass that information on.
And Leonore, we got time for one quick question.
Oh, I just wanted to follow up on that too, because we've also had conversations with Mary Williams, who runs the teaching tools in plant biology for ASPB, about possibly including that in the teaching tools, and a couple of other people who have expressed interest in including the curriculum. So that's sort of like the place that it needs to go next, is the dissemination and uptake.
Yeah, I suspect once it's out and people can see it and review it themselves, we'll get more pickup.
All right, that brings us to time. Thank you, Megan. And we'll move on to our next presenter. Tim Cezard is presenting on behalf of the Standards for Genetic Variation Working Group. So yeah, take it away, Tim.
Thank you very much. You can see my slides. All right, so I'll be presenting what we've been working on for the past year or so, with a little bit of a reminder of what we've been doing before. But first, a little bit about who we are. So we are a very diverse group that covers a lot of the different type of database and resources that deal with variation data. And that goes from myself, where I work at the European Variation Archive, or aggregators that will deal with bringing data together, or community database that are much more focused on one particular species, or members of organization that will provide identifiers for things like plants. So we're very diverse, but we're always looking for new members. I've tried to list the currently most active members at the moment. There's people that come and go, but we're always looking for increasing that diversity. In particular, we have a big focus on plants at the moment. We would like to expand that to other animals as well. Right. Our goals, they've evolved a little bit over time, but they're still on the same theme. So we want to improve availability and reusability of variation data sets. You'll see why in a minute. We are actually doing this bringing together of different people from different parts of the community, data providers, curators, and people like me that are more computer scientists, to promote the interoperability between the different resources that we organize. And lately, we've had a big focus on using standard formats, but also standard identifiers for both data and metadata, and I'll explain that in a little bit. All right, as in lieu of a landscape analysis, there's been a massive amount of work done by my co-chair Marcella was started last year by essentially trying to curate a large number of papers that had variation data. And job was to try to retrieve that variation data and try to associate with all of the important metadata, things like the reference genome that they were associated, the actual original sample they were derived from, or just get the actual variation data. And that was a massive endeavor. And it taught us a lot about the issue that are facing these kind of data sets in the wild, is that when they are published, a lot of the data sets are either unavailable completely or only available at the raw data, or available at different levels. So she really tried to push the original author to submit their data to the relevant database with some amount of success, but not all of them actually did so. But that gave us really the foundation for having a better understanding of what I call data journey for these variation data sets, which is essentially when the data is published, when the paper comes out, where does the data go, and how can it reach the person that actually wants to reuse that data set. And there's different level of the different paths by which these data travel. And the most common is the first one, is when the one that is actually mandated by funders is that the raw data is made available to the community. And that's what most people do, and that's what most journals require. And so when you have raw data being made available to the community, that's a very good start, but there's a massive amount of curation required to be able to actually recreate the variation data sets when you start from the raw data. And there's often a decorrelation between, but it's not always easy to go from the raw data and reproduce exactly what was done in the publication. So that means that the data set that you can get at the end might actually be different. But that's the main thing that is actually required by funders at the moment. The second case that we've encountered is when the variation data has been made available, but it's been made available in a generic, in a generalist database. That could be supplementary data, or that could be deposited in places like Figshare or DataDryad. So those are definitely an improvement of the first case. So the data is then available for people to reuse. And so it prevents us from having to actually request data from the original author. But these generalist databases, they don't necessarily have the knowledge to be able to check prior to publication that the format has been respected, that the data are correctly linked. So for example, that you have an associated reference genome for these variation data. And so that's really an improvement, but it's not quite perfect. And the last one is when the data is actually made available to either a specialized database like the European Variation Archive, or a community database that would actually take care of doing this work of creation before the publication is actually done. And this is definitely the best possible journey, because the data is then readily available, but also you have much richer metadata, and the actual format of the original data has been checked. So these kind of landscape analysis allowed us to land on specific standard that we wanted to highlight and promote. And for variation data, the main format that is being used at the moment is the variant call format, which is a file format. And that's what the European Variation Archive provide and receive. But to be really relevant, it needs to be associated with a genome that has been archived as well in a database that provide long-term accessioning like INSDC. But again, the important thing is not necessarily just to have a VCF file, but to have a VCF file that you've been able to validate. So you've been able to say that it actually follow the format that has been specified. And for the metadata, we've been working mostly with biosamples. So biosamples are provided by members of the INSDC, so that could be NCBI or EBI. They both have resources that will provide, and also is DDBJ actually. And we provide biosample, and they have the advantage of being able to aggregate any amount of metadata. But that also mean that they don't necessarily have a lot of requirement. So we've made recommendation alongside some colleagues from Elixir on what they should contain. And we have kind of coalesced around a community-defined checklist that follows MyEpi standards. And so, yes, so we've relied on these recommendations that have been made by colleagues from Elixir, both on the VCF file. They've published a paper in 2022 that made very good recommendation on how they should be the VCF should be formatted so that it can be easily the data can be easily extracted. And the metadata associated can be easily linked. And they also have published a step-by-step guide on how to validate your data so that it can be taken directly by these database. So it can be submitted very easily to biosample and EVA. And talking about the submission to EVA, so that's an example of what the workflow post-submission actually does. And so you could see that there's a large amount of work that is done at the validation level. So before any submission is accepted, there's a lot of steps that will check whether the format of the initial file is correct, whether the metadata contains enough information, enough of the relevant information. And we then do cross-linking between the submitted data and the relevant database so that they can the submitted variation data can be cross-linked with the reference genome, but also the sample that have been associated with the variation provided. And that's where we recommend the usage of green global and other germplasm identifiers so that the original sample can also be identified. Once the data has been archived in the EVA, one of the main function of the EVA is to provide variation identifiers. And so what these variation identifiers allow you to do is to have specific names for one particular loci on the genome that we have evidence, for which we have evidence that it's variable. And that's been a very successful identifier, at least used in human. So these variant identifiers, they're globally unique. So that means that there should only be one for one location. And they are long-term, which means that even if they will always be available from the resource that they've been emitted from. And they identify a loci on the genome, and they are stable across genome assembly version. And what that means is when a new assembly version comes along, these are remapped to the new assembly version, and the same name is kept. And so they become a very convenient identifiers for aggregating relevant information for variation. So any information that can be associated with a variant can be associated with those identifiers. And that's what happens in human, at least. So you've got large projects that have created population allele frequencies, and those have been associated with RSIDs. And you've got large projects that have provided QTLs or EQTLs like GTEx, and they also associate these with RSIDs. You got GWAS results or clinical traits, and these are again associated with those RSIDs. So we decided that it was a very good example of an identifier that we should promote within our group. Oh, another thing that I forgot to mention, there are existing machineries and infrastructure that have already been developed around these identifiers, mostly for humans, but they are reusable. They already exist. And one of these, for example, is that any paper that contains an RSID would automatically be searched by a resource called Europe PMC, and so they can find these papers based on the usage of these RSIDs. So you can search by RSIDs and find all of the papers that have this RSID included in them. And they've got about 1 million publications that have RSIDs in them. Most of them are human, but what we're hoping for is that as they are becoming to be more prevalent, they can be used in other species as well. So what did we do? Oh yeah, sorry. Yeah, so in non-human, they are being used in many aggregators or many genome browsers mostly at the moment. So they're really starting to be kind of used in other resources. So you can find them in Ensemble, you can find them in UCSC, you can find them in NCBI Genome Data Viewer, and you can find them on other resources. And I'm probably missing some here. But so what did we start to do? We started to make sure that community databases that are using variation data are using those RSIDs as identifiers for those variation data. And there's been a massive amount of work done by the Gramine and Sorghum base, by Marcella and Sharon, of integrating the release 5 of EVA into Gramine and Sorghum base, with quite good success actually. So most of the variant could be annotated with RSIDs, and so you get several millions of variants that are now from Sorghum base that are now associated with known RSIDs that can be linked back to EVA to find all of the other project that contains this that reference, that variant. That's a very successful project.
Hey, Tim, we're running short on time. How much do I go?
Okay, I'll speed up a little bit. Another example of those was at Soy-based product led by Rex, which just brokered about 4.8 million variants to EVA, and we've just released these 1 million new RSID, and that should be available to the public in our 6th release. And the last one is we are starting a new project with industry partner where we are providing these RSIDs so that the SNP arrays can be annotated with them. And we are setting up a process by which the RSIDs can be requested from the industry partner so that we can provide them a full set of RSIDs. And that's been done successfully in one project with Agriplex, where 2.4 thousand SNP have been marked with the release, and 26 new ones that were missing RSIDs have been added. And we're currently leveraging the group's contacts to work with Timo Fisher. And we are actually open to any other contact that you might have. Just to summarize, we've done a landscape analysis trying to verify a number of studies, and that really allowed us to identify the data journey and better understand the creation challenges. We worked with, in our first year, we worked with our metadata group and we standardized on the germplasm identifiers. And lately, we've been working on promoting these RSIDs in community-based data aggregators and industry partners. And at the moment, we're writing our white paper to kind of summarize all of these results and make them more publicly available. I also wanted to give one word on the fact that we've also merged with the public genomic resources. And that's a recent addition and led by Maura. They are bringing new challenges to our group. So things like identifying haplotypes instead of variants, or where to store them. And so that's also something that we will tackle in the next months, next 6 months or so. And I'll skip this breakout question. We'll get to that in a minute. Thank you.
Awesome. Thank you, Tim. We're over time here a little bit, but I want to offer a chance for quick questions or comments from anyone. All right. Thank you, Tim. And yeah, so we'll get back into it with the reports from the breakout rooms. These are going to be 5 minutes each. So try and keep it short. I know at least in the breakout room I was in, we had we covered way more than could possibly fit in the 5 minutes. So yeah, hopefully we can get just the summaries here with the notes that'll be posted later. Yeah, so we'll get started with Education Breakout Room 1. I think, Meg, you were going to present?
Yep, that's me. So we had a great discussion. I think the most important thing to share with the full audience is that we would really like to see some beta testing of our material, get in there, poke holes in it, see where you find gaps, see where you think it needs more depth. We're really ready for that at the undergraduate and graduate level for those of you who are teaching faculty. And if you're not, even if you're here representing a database, you still have maybe user training. So if you want to adapt some of the material for user trainings and give us feedback, that would be super. After beta testing, when we're all pretty comfortable that we have a good product, how do we get it out there? Thinking we need a YouTube channel, link to the main AgBioData YouTube channel, linking to all of the slides and stuff, and maybe have an AgBioData monthly webinar for the education group to kind of advertise when we're launching. We hope to do online and in-person versions of the curriculum, but looking for a new funding source for that to support people time. And we talked a little bit about doing an OER Commons book and how feasible that might be. High school teachers were an originally big target for us, but we currently don't have a champion on the working group who knows a lot about high school education. So if you have a high schooler or if you know high school teachers and you want to be that champion, we'd love to work with you. But for the most part, we're going to focus on where we can have a high impact, which appears to be more the university collegiate level. And for new material, we got a lot of great suggestions. Talk about data federation, talk about publishing for fairness, biocurator module, thinking about how AI is going to be used for biocuration. So I think it really, the overall theme is we need to be working with other working groups to get their outputs incorporated into the educational curriculum. And so that about summarizes our discussion. Thank you.
Wow, that was super fast. Thank you. Any other members of that breakout room? Any other comments? Anything missed? All right, cool. Thank you very much, Meg. We'll move on to the second education breakout room. Who's going to present for that one?
Thanks. I will. I'll make it
Oh, John, you're muted.
myself. So I'll make it even shorter. Everything that Meg said, we agree with. We had a smaller group of a diverse group, nature of people, so we didn't get into a lot of that level of detail. But we agree it's time to get it out there. We've got a white paper that's all but done. We need to get out there. And so that's the bottom line. We need to move this forward. I think there's a lot of applications. One of them that you did mention, Meg, is, the requirement coming up for public use of databases. And also, in response to your high school thing, yeah, I've kind of worked with high school educators pretty closely for the last 30 years. And data management and computer science are the thing, really, for a lot of them. So it's something that we could talk about for sure. And that's it.
Awesome. Thanks, John. Any other members of that breakout room have comments or anything else to add?
I'll just mention that I think having I'm not a teacher. I work for the USDA, so I'm not allowed to teach, but like I guest lecture. I think everybody could do a little bit to promote this work to kind of help, because I know the education work group has put a lot of effort into all this, and it is a great labor of love, and I really want to assist the working group where I can. That's all.
And there were some kind of concrete suggestions in terms of when we're ready to prominently promote the curriculum on the AgBioData website, and with an appeal to educators to incorporate it into their curriculum. There was also a question about, when we're onboarding new AgBioData members, can they be made aware of the availability of the curriculum? WSU has, I think, a 10 course series that's either being developed or refined. And there's a postdoc who's involved in that process who could be invited to the working group. And as one of our participants has that connection. And then whenever we're ready to promote it more broadly, I'm happy to share with agricultural librarians who may be able to promote the curriculum across their campuses.
Yeah, thank you, Layla. And we did talk about that level of detail. So thanks for bringing that up.
All right, cool. Any other final comments for that breakout room? All right, excellent. Thank you. And we'll move on to the final breakout room, the genetic variation group. Who's going to report on that?
It's me, Marcela.
Okay.
Okay, so we tried to fill in your beautiful template, Narita. So question one was, what are, if any, the barriers for submitting variation data to a central community or community database? Let me put in a slideshow. So the discussions, we identified several limit several issues that people found. You can read them on the left, and really some of the recommended actions that we proposed is we're suggesting we need biologist friendlier tools to validate metadata, especially for NCBI repositories, because for genetic variation, one of the prerequisites for submission to a repository for variants to the EVA is precisely that the assembly has been submitted to one of the INSDCs, and at least in the US, NCBI is one main repository, so that's usually like a bottleneck. A lot of people have encountered problems with that submission, so suggest better documentation. Especially making the informative error messages where there is no validation to validate the data, community databases where there exists a database for that community, say maize or sorghum or Arabidopsis, to have those databases to act as brokers for uploading the data has been validated to EVA. Work with the community database for these uploading tools. Also, one interesting suggestion was to have a national database brokers that help that data to go into EVA, and say data that is coming from like a Chinese databases, like there is a Chinese broker that helps all the databases or the different groups to submit to EVA and to have, because they will have preferences to use the germplasm repositories with those identifiers or things that are more like regional specific. So on one hand, we have the species specific databases, but on the other might be like regional databases, not only national, because it could be Europe. And with the examples of successful submissions. So those were some of our suggestions. And another question was, interestingly, not everyone that joined our group had been aware of RSIDs before, and they came to this discussion after hearing about RSIDs during Tim's presentation. So that was another interesting point. So outreach becomes very, very important education. So the next question was what metadata would be required for you to make the genomics variation reusable and interoperable. So there exists the lack of validation tools. Was brought up because often, and it's hard because every community has its own standards. So like a major repository like EVA may not have that expertise on specific species communities. So it's hard to do that with tools. So it still needs a lot of manual creation, and that's where the brokerage from the community database comes in place. It's important to have, we stressed standardized germplasm identifiers, but also synonyms preserved, and a lot of details, proper details like passport details for the germplasm, to make that data as most usable, to be linked to like traits, and to have more informative for when it comes to like breeding, come up with markers that are that's going to be used in breeding. So some of the recommended actions: community databases that can be brokers, more community checklists and examples for users that help users complete it, specific documentation, repositories should try to, like NCBI, should lower the barrier for submission to identify some of the most problems. Can be identified in FAQs, but they are more useful at the beginning when people is actually creating their data, designing their experiments. So it might be useful to have more some documentation, some outreach explaining what you're going to need, or why is it important that at this point you have standard identifiers in place to avoid further problems later and facilitate things. And some successful examples, things like that. So I'll stop sharing.
Awesome. Thank you, Marcela. Are there any other comments or thoughts from that genetic variation working group breakout room? All right, excellent. And with that, I'll turn it over to Anna-Rita to finish out our day.
Well, yes. So thank you again for everybody that joined today. It's just a reminder that tomorrow is the second day, same time, same Zoom link, and we will have the Data Federation training with Peter presenting, and two other active working group, the new one on single cell RNA, sick biocreation, and the sustainability working group. So thank you so much, and see you all tomorrow.